study mouse sperm stored in space produces baby mice on Earth. Japanese researchers have successfully produced healthy mice from frozen sperm that was stored on the International Space Station ISS for almost 6 years. The sperm was freezer dried and sent to the ISS in 2013. Freeze drying is a process that removes water from frozen substances in an effort to make them last for a long time. Once the sperm was brought back to Earth and unfrozen, it resulted in the birth of 168 healthy baby mice. The researchers said there were little difference between the mice produced by space sperms and those made with usual sperm. Teruhiko Wakayama is a developmental biologist at Japan's University of Yamanashi. He was the lead writer of a study reporting the results in the Science Advances. All pups had normal appearance. Wakayama told the French news agency AFP about the experiment. When researchers examined genes of the mice, no abnormalities were found, he said. The team launched 3 containers, each carrying 48 samples of freeze-dried sperm to the ISS in 2013. Freeze-drying permits substances to survive at the room temperatures for up to a year, so no freezers was needed on the launch rocket. On board the ISS, samples were kept inside the freezer at minus 95 degrees Celsius. Samples were returned to Earth at different times. The first were flown back after 9 months, and the second after 2 years. When the last containers returned to Earth in June 2019, it had spent 5 years and 10 months in the space. The returned samples were injected into egg shells, which were put inside female mice. The experiment aimed to find out whether talking in radiation for long periods in space would damage DNA in reproductive cells or pass on mutations to babies. These same issues could be a problem for humans in the future if space exploration greatly expands, with larger populations possibly living on other planets. Deep space is filled with high radiation produced by solar particles and cosmic rays from outside our solar system. The researchers said the sperm cells experience radiation levels 170 times greater than sperm kept in ground storage for comparative purposes. The researchers reported that the spatial radiation did not damage the sperm's DNA or reduce its fertilization ability compared to the sperm caps on Earth. In the future, when the time comes to migrate to other planets, we will need to maintain the diversity of genetic resources. Wakayama wrote in the Bible on the experiment. He added that structural diversity both in humans and animals can protect against increase in harmful mutations seen in inbreeding.